Good morning all of you. My name is Janvi from BSc Chemistry Honors Final Year. Today we are going to discuss the topic ion exchange chromatography. Chromatography is the separation technique to separate the ions component from the mixture of components. Purification is done on the basis of ions in ion exchange chromatography. Any charged molecule like protein, nucleic acid or any other charged molecule can be separated with the help of ion exchange chromatography. For the instrumentation of ion exchange chromatography, a mobile phase is attached with the pump and the pump is attached with the column. The column is further attached with the detector and that is further attached to the computer on which we get the graph. Column is a tube-like structure which is attached with detector and detector is attached with computer which gives us final graph. First step is in starting bottle have mobile phase in the column form of the buffer solution at fixed pH. It is called mobile phase because we mix the sample in this buffer solution and then reaction into the column. Mobile phase helps in the movement of sample over column, that's why it is called mobile phase. There is a stationary phase present in column. Solid absorbent material which is called a stationary phase. In case of ion exchange chromatography, stationary phase solid absorbent material is made up of resin. Resin have very small beads which carry charge. Now on the basis of resin, ion exchange chromatography is of two types. First is the cation exchange, second is the anion exchange. If beads carry negative charge, then it is called cation exchanger. If beads carry positive charge, then it is called anion exchanger. In an ion exchanger, beads have positive charge, functional group is negative charge. Resin have some modification that is resin is attached with the functional group which carries negative charge. In cation exchanger, resin is negative charge and the functional group is positively charged. Resin beads have negative charge and the functional group is positive charge. Different type of resins are used in anion exchange chromatography. Resin are categorized strong exchanger and weak exchanger. Now, what is the principle of an ion exchange chromatography? How purification is done? First of all, we take the sample having different components like A, B, C, which is mixed with buffer solution having specific pH. Now, sample is ready to run on the column. Suppose sample that is protein having negative charge. Sample in the form of protein having negative charge is going to load on anion exchange column. Now, anion exchange resin have negative charge functional group. There is a competition between protein negative charge and functional group negative charge, that which will bind with positive charge resin. Now, protein negative charge will bind with positive charge resin and replace the negative functional group. Now what happened? Protein negative charge exchanged the functional group negative charge from the positive charge bead of resin. So there is exchange of anion, therefore it is called anion exchanger. Case 2, if sample that is protein is positive charge. Now there is exchange of positive charge on the negatively charged bead of resin. Now, how charge of protein is decided. Protein is made up of amino acids. Some amino acids carry positive charge, some carry negative charge. So, overall charge of amino acids by which protein is formed will decide the charge of protein. And on the basis of overall charge of protein, we will decide we use anion exchanger or cation exchanger chromatography. Thank you.